Let's say we have the part of the amino acid sequence of some protein, or we have antibodies associated with that protein. And what we would like to do is we would like to identify the DNA associated with that gene, isolate the gene, so we to narrow it down to find which DNA it is that codes for that amino acid sequence or the protein for which we have antibodies, and then make large number of copies of that gene's DNA. And once we have done that, we have isolated the gene, and we can do further experiments like characterize the DNA sequence of the gene or how the gene works, and perhaps use it for genetic engineering applications like transgenesis, or making recombinant protein. And genomic or cDNA libraries and screening is one way of achieving that goal. Another way is polymerase chain reaction, where you have primers that can amplify a given gene in a specific manner so that you only copy the DNA associated with the gene, and not anything else. And making these genomic and cDNA libraries and then searching through them or screening them to identify the gene of interest is another way of achieving the goal of isolating a gene and getting large numbers of copies of that gene's DNA. And the way this approach works is something like this. So let's say we have the genome of an organism, and it contains several genes shown here in green, purple, and yellow. In the first step, we break up the DNA of the genome into different pieces. So maybe we have a piece that contains the green gene, and maybe we have a piece that contains the purple gene, and another one is yellow. And of course, we are going to not have just a few handful of pieces. In reality, we are going to have tens of thousands of pieces of the genome. So the first step was to break up the genome. And once you have broken up the genome, then you use molecular cloning techniques. So the cutting and pasting of DNA into vectors to create thousands, tens of thousands of different vectors, each one containing a different fragment of genomic DNA, and some of them are going to have one gene, another one is going to have a different gene, and so on and so forth. And so this is thousands, or let's say tens of thousands of vectors or plasmids. And these tens of thousands of vectors are not sort of in separate tubes. They are all together in one tube, in one solution. You have tens of thousands of vectors, and that's why this is called a library. This is a library of DNA fragments, and since it's coming from the genome, it's a genomic library. Okay, and then in the next step, you search this library. And the word biologists use for searching is screening, which is basically you use the information such as the amino acid sequence or the antibodies against the protein that you have. You use that information to identify which of these plasmids among the tens of thousands of vectors in your library, which of them corresponds to the amino acid sequence that you have, or the protein whose antibodies you have. And then once you have identified this gene through screening, or the DNA corresponding to the gene through screening, you can further amplify, since these vectors are actually in the process of molecular cloning. Each of these vectors is inside bacterial cells. You can simply grow the bacteria that contain the vector that contains the DNA corresponding to your gene, and obtain large numbers of copies of the gene's DNA. Now, before we go into the details of how this process works, let's discuss the different vectors that one can have. And we've already discussed one type of vector, which are bacterial plasmids. And bacterial plasmids, they are episomal DNA that contain an origin of replication that allows them to be copied independent of the bacterial chromosome. Another one is bacteriophage vectors. And in bacteriophage vectors, you package DNA inside a bacteriophage, a virus that infects bacteria. So when you infect the bacteria, that process is called transduction. The virus, the bacteriophage, enters the cell, and the bacterial cell, and since its genome contains the DNA that you want to introduce into the bacterium, you end up introducing that your DNA of interest into the bacterium. Another kind of vector is called a fosmid, which is a hybrid of DNA from the fertility factor, or F plasmids, and from bacteriophage lambda DNA. And these plasmids, they are delivered as vectors through transduction to bacterial cells. But once they're inside the bacterial cells, these sites, cohesive sites or COS sites, cause the viral DNA to circularize. And since the viral DNA or the plasmid DNA carries the origin of replication, this will then start replicating in bacteria along with the bacterial chromosome. Another one, which is also based on the F factor origin of replication, is, or the based on the F plasmid, is bacterial artificial chromosomes. And what's special about both fosmids and BACs is that even though plasmids can carry inserts of up to 10 kilobases and bacteriophage vectors can carry inserts of up to 15 kilobases, fosmids can carry much larger DNA inserts, so up to 45 kilobases, whereas BACs can carry inserts of a much larger size, 200 kilobases. And this is important because the human genome, for example, is 3 billion base pairs long. And if you were fragmenting the human genome and cloning each fragment into plasmids, you would need a very, very large number of plasmids. So your library would have to have a very large number of plasmids. Whereas working with BACs, since each BAC can have a large DNA inserts of up to 200 kilobases, the size of your library, or the number of different vectors that you have, is 20-fold smaller, and that library is much easier to work with. Now, how do you get these vectors into bacterial cells? As we have discussed, plasmids, it's a transformation, the process by which bacteria can take up extracellular DNA, and then you can grow these bacteria and get a bacterial colony. Fosmids are packaged into bacteriophage genomes. So you infect the bacteria, and that process is known as transduction. But once the fosmid or the bacteriophage is inside, the cos sites cause it to circularize, and you now again, similar to a plasmid, you get a circular episomal piece of DNA that replicates with the bacterial chromosome, and you get bacterial colonies. Unlike fosmids that lack the viral DNA replication machinery, bacteriophage vectors have all the machinery required for infection. So for the virus particles to enter the bacterial cell, but also to replicate inside the bacterial cell, and then to break open the bacterial cell, which is called lysis, and to form a plaque. And when you use bacteriophage vectors, you basically detect the DNA in these plaques. So there are different vectors, and depending on the application, we may choose different ones. If the DNA insert is small, we may choose plasmids or bacteriophage vectors. But if our inserts are big, such as when we are making a genomic library, then we may want to choose BACs or fosmids instead. So the next question is, once we've made a choice of vector, how do we break up the genome and use molecular cloning to make a library of vectors? Now, as it turns out, restriction enzymes make this a one-step process essentially. So let's say we have the genome, which is the donor DNA or the DNA that's going to be inserted into our vector, and we have plasmid. Then we can digest with restriction enzyme. So in this example, we are digesting with EcoR1, and recall that the recognition site of EcoR1 is GAATTC, and it's palindromic. Now, in any given genome, this pattern of sites will occur by random chance at some frequency. And for this EcoR1 site, it is going to be about 1 in every 4,000 base pairs, roughly, that this EcoR1 site will occur. So if we take the entire genomic DNA and treat it with EcoR1, it's going to cleave in several locations once in every 4,000 base pairs, and we will end up with genomic DNA fragments with an average length of about 4 kilobase pairs. And then we can mix the digested vector, the digested plasmid, as well as the digested DNA. And in contrast to regular molecular cloning, where you have just one fragment that you mix with the vector, here we will have tens of thousands of fragments that came from the genome. And we will mix all these fragments with the vector and then treat with DNA ligase. And when we treat with DNA ligase, the DNA ligase will make the phosphodiester bonds between the G and the A, and create a contiguous circular plasmid that contains the insert, except now, since we started with tens of thousands of fragments created by EcoR1, we will have tens of thousands of vectors, different vectors. The blue part of the vector is going to be the same in all of them, but this orange part is going to be different. It's going to be different DNA sequences, depending on which part of the genome randomly got ligated into that particular plasmid molecule. And we can roughly estimate how many vectors, or what is the size of the library going to be. So let's say we were using BACs, then the human genome is about 3 billion base pairs, and a typical BAC will carry 200,000 base pairs, and that corresponds to 15,000 vectors, or 15,000 BACs. If we wanted each fragment of the human genome to appear once on average in the vectors, usually, since this is a random process, you want a library that is bigger than 15,000 in order to make sure that every fragment is cloned in at least once. Now, the fact that genomic libraries are so large makes it difficult to work with them, and it's possible to make a different kind of a library called a cDNA library that is much smaller. And the rationale for a cDNA library goes like this. We are most of the time only interested in the coding sequence of the gene. Now, coding sequences form only 2% of the human genome, and this is generally true for most organisms. And therefore, instead of looking for a gene in 3 billion base pairs, why not look through in only 2% of that, which is 60 million base pairs? So if we could make a library only out of the coding sequences of the genome, then our library would be much smaller, and our job of screening will be made much easier. It would be 50 times easier, because the library would be 50 times smaller. And it's possible to make such a library by taking the RNA, all the expressed messenger RNA in a particular cell type or a tissue, and then converting that mRNA into DNA. Now, recall the central dogma, which is that DNA gets read into messenger mRNA through transcription. And if we could reverse this process, which is we could reverse transcribe mRNA into DNA, then we would have only the coding part of the genome, and not all the other parts, the non-coding parts of the genome that we are not interested in. And it's possible to do this reverse transcription using an enzyme called reverse transcriptase. And this enzyme was developed by, or evolved by, retroviruses. For example, HIV or the coronavirus, whose genetic information or genome is in the form of RNA, and in order to use the cell's DNA replication machinery, these viruses convert their RNA genome into DNA using this enzyme reverse transcriptase. And we can use this enzyme isolated from these retroviruses to do the same. And the process goes something like this. So let's say we have a gene in DNA. And so this dark blue is DNA. And this gene will have exons, which constitute the coding sequence of the gene. And it also has introns. And then this gene is transcribed in the cell to create a pre-mRNA, which contains the exons and the introns as well. And this pre-mRNA is processed by the splicing machinery to remove the introns, and subsequently a poly-A tail is added to the RNA. And so in order to isolate the DNA of this gene, what we can do is we can take this RNA, isolate RNA from cells, and then you use what's known as an oligo DT primer, basically a bunch of thymines, and that acts as a primer to start the reverse transcription process using the reverse transcriptase enzyme. And what the reverse transcriptase enzyme will do is, just like RNA polymerase uses Watson-Crick base pairing to build an RNA molecule using a DNA template, a reverse transcriptase will use RNA as a template and Watson-Crick base pairing to build this DNA molecule, a single-stranded DNA molecule. And since this DNA is complementary to the mRNA, it's known as complementary DNA or cDNA. And so, strictly speaking, in this process, we are not making DNA, we are making complementary DNA, cDNA. And then this reverse transcriptase enzyme makes a hairpin loop and stops, and then you can use a DNA polymerase to make the other strand to get a double-stranded DNA, and then you remove the hairpin and end up with a double-stranded complementary DNA, which corresponds to the coding sequence of the gene that produces the final processed mRNA. And sort of the things to keep in mind about cDNA is what it does not have. cDNA will not have things like promoters, which are never transcribed, or enhancers, which are non-coding sequences. It will not have introns, and it will not have the poly-A tail. So these are things in the DNA or mRNA that the complementary DNA will not have. What it will have are exons and the coding sequence of the gene. Now, we use this example where we started with only one mRNA molecule. However, it's possible to do this process in parallel if you take all the mRNA in the cell. So let's say there are 5,000 genes expressed in a particular tissue. Let's say we took brain tissue, and there are about 5 to 10,000 genes that are expressed in brain tissue. And if we treat that RNA with reverse transcriptase and a DNA polymerase, we will get 5,000 or however many mRNA molecules there are. We'll get a corresponding number of cDNA molecules. One thing to remember when we are building a cDNA library or when we are doing reverse transcription, is that we will not end up with the DNA corresponding to a gene if the gene is not expressed in a particular tissue. So it becomes important when we are doing reverse transcription to build a cDNA library to appropriately choose the tissue to extract RNA from, so that the gene that we are interested in is actually expressed in that tissue. Usually this is not such a big problem because we know something about the biology of the gene. So for example, if we were trying to isolate the human growth hormone gene, we know that HEH hormone is secreted from the pituitary gland, and therefore we should use RNA from the pituitary gland to isolate human growth hormone. If we instead try to use RNA from the pancreas, it would not work because the HEH gene is not expressed in the pancreas. Now, the next step to making an actual library is to take this cDNA and keep in mind that here we have thousands of different cDNA molecules, each corresponding to one gene expressed in whatever tissue we got the RNA from. And in the next step, we take this collection of cDNAs and clone them into a vector. Now, these cDNAs are not going to have EcoR1 sites at the end, so we must add the EcoR1 sites or whatever enzyme we happen to be using. And you can do that by blunt cloning, where you basically have blunt-ended DNA fragments and you use DNA ligase, which will make phosphodiester bonds, and just add these flanking sequences that contain, in this case, EcoR1 sites, but it could be any restriction enzyme. And then you can digest these complementary DNA, cDNA molecules with EcoR1 and take these cDNA molecules now with sticky ends and ligate into a vector that has also been cut with EcoR1, and you will end up with a cDNA library which will have thousands of bacterial cells, each with one unique vector, and that unique vector will have one complementary DNA inserted into it, so that you will have one vector or one bacterial cell per gene that is expressed in the tissue that we make the cDNA library from. Now, the final step, once you have made either a genomic library or a cDNA library, is to then find the clone or find the bacterium that contains the vector that contains the insert corresponding to your gene. And that process is called screening. So you can screen at least two ways. And one way of screening is that, let's say our gene, it has a sequence and we have figured out part of the sequence based on the amino acid sequence of the protein encoded by this gene. Now, if we know a fragment of the DNA sequence of this gene, then we can design a probe that's complementary to the sequence. And it's called a probe because this DNA molecule is radioactive. So you have either used a radioactive isotope of carbon or nitrogen to make this DNA, or you have attached some kind of a molecule to this DNA that fluoresces, and we are able to see wherever this probe is because of the radiation given off by the radioactivity or fluorescent light emitted by the probe. Now, we can screen the library in the following manner. We can take the bacterial cells of our library and plate them and let them grow overnight. And when they grow overnight, they will form these colonies, each colony being cells that are descended from a single clone, and therefore all the cells in the colony contain the vector corresponding to one DNA or cDNA fragment. And then we can transfer these colonies to a nitrocellulose membrane. So you basically lay the membrane on top of the plate so that some of the cells are transferred onto the nitrocellulose membrane, and the pattern of the colonies on the nitrocellulose membrane will be exactly the same as the pattern of colonies, how they're laid out in the plate. Now you take this membrane and you incubate this membrane with your radioactive probe. The radioactive probe will go and bind to those colonies that contain the DNA sequence that's complementary to our probe sequence. That is, the probe will only go and bind those colonies that that contain the gene that we are interested in identifying. And then you take this nitrocellulose membrane and you wash away, you wash it in water in order to wash away any probe that's not bound. So that will only leave probe that is sticking tightly through hydrogen bonding to the DNA of our gene, and therefore only those colonies will contain the probe that contain the gene that we are interested in. And then you take this nitrocellulose membrane and you expose a photographic film with it. And if you're using a radioactive probe, it will emit radiation that will create a spot on the film in the location where the positive colony is located, and we can use that location to look in the plate and find the bacterial cells that contain the gene that we're interested in, and you can pick those bacterial cells with a needle and then grow them up in a big culture in a large amount of medium to amplify the gene that we are interested in. And of course, if this were not a radioactive probe, but if we had attached a fluorescent molecule, then instead of simply exposing it to film, we would probably excite the fluorescent probe with light and then image the nitrocellulose membrane. But the principle remains the same. We will be able to see through the light emitted from the probe which colony is positive. And this is the one type of screening with DNA or RNA probes. Another type of screening involves using antibodies, and that's a situation where we have an antibody against the protein encoded by the gene that we are interested in, and you use fluorescently tagged antibodies, secondary antibodies, to identify the colonies that are positive for the gene of interest.